Hello everyone, today I will present the work FastFold, reducing AlphaFold training time from 11 days to 67 hours. The protein folding problem, also known as protein structure prediction, is one of the most important topics in the field of structural biology. The central dogma of molecular biology was proposed by Crick in 1958. DNA makes RNA, RNA makes proteins, and proteins in turn assist the first two processes and help DNA to replicate itself. According to the Inference dogma, we can get the protein structure from the gene sequence. However, the multi-level structure and the complex interactions of the proteins make it challenging to accurately predict the three-dimensional structure. Protein structure prediction has been an important research problem in structural biology for more than 15 years. Predicting the three-dimensional structure of proteins directly from their amino acid sequence has a wide range of applications in many fields, including drug design, protein design, etc. Prediction of protein structure is generally divided into two methods, experimental and computational. Experimental methods can obtain more accurate protein structures, but they require more time and economic cost, while computational methods can obtain high-throughput protein structure prediction at low cost. A large amount of sequence data has been accumulated in biology, but protein structure data is still relatively small. Therefore, it is very important to improve the prediction accuracy of computational methods. The recent success of deep learning has led to a wide range of applications in various fields. Convolutional neural networks has been introduced to protein structure prediction by Rap2X and AlphaFold 1. These CNNs achieve significant performance gains in the critical assessment challenge of protein structure prediction, also known as CASP. These works demonstrate that deep neural network can be an effective solution to the challenge of protein structure prediction. At the end of 2020, DeepMind announced a new generation of AlphaFold, which successfully introduced transformer model architecture to predict the 3D structure of protein directly from sequence end to end, and it achieved a huge performance improvement at the CASP 2020 competition. AlphaFold is also the first to achieve atomic level accuracy in protein structure prediction. AlphaFold's success has attracted significant attention from the scientific community, making it one of the most influential scientific achievements of 2021. It was selected by Science magazine as the scientific breakthrough of the year, and also Nature magazine has named AlphaFold as one of the science news that shaped the year. The recognition is the result of the effectiveness and potential impact of the AlphaFold approach on solving the protein structure prediction problem that has been a long-standing challenge in the field of biology. Let's first look at the main model structure of AlphaFold. AlphaFold is an end-to-end model, which means that the input of the model is the amino acid sequence, and it directly outputs the 3D structure of the protein. AlphaFold mainly consists of an embedding module, AlphaFormer module, and a structure module. The amino acid sequence is encoded into MSA representation and pair representation after the embedding module, then processed by 48 AlphaFormer blocks, and finally the highly processed representation is fed into structure module to get the three-dimensional structure of the protein. Because the AlphaFormer architecture differs from the standard transformer, its two-dimensional representation information posed a significant challenge for the training and inference of AlphaFold. With an additional sequence dimension, not only is there an order of magnitude increase in computational complexity, but also an order of magnitude increase in its memory consumption. In general, there are several challenges in the training and inference of AlphaFold. The first challenge is the time and economic cost. DeepMind used 128 Google TPU v3 to train AlphaFold, which took up to 11 days to complete. The total TPU time consumed was over 30,000 hours, making the research related to AlphaFold very challenging due to the significant time and cost required for training. The second challenge is the huge memory consumption, which exceeds the memory capacity of the most GPUs. It takes 20 GB to store all the activation of attention context, and it is impossible to save all the activations in the training. And there is a huge challenge of long sequence inference. Inference of longer sequence required much more GPU memory, and for the AlphaFold, the inference time for a long sequence can even reach several hours. In FastFold, we propose a parallel AlphaFold architecture with low-level optimization technologies, including communication and computational optimization. The dynamic axial parallelism is proposed based on the characteristics of AlphaFold. It differs from traditional tensor parallelism by dividing the activation along the sequence dimension and using all-to-all communication, which allows training to scale to more computational resource, thus reducing the overall training time. To further reduce the communication overhead in a cluster environment, we propose the duality ASIC operation method to overlap computation and communication during training. We also implemented several computational optimizations, including fused kernel and rewritten kernel, which significantly reduced the training and inference time of AlphaFold. For training, FastFold reduced the overall training time from 11 days to 67 hours and provided significant cost saving. Additionally, FastFold was scaled to 512 NVIDIA A100 GPUs, achieving a total of 6.02 petaflops. For inference, FastFold achieves a speed up of 7.5 to 9.5 times for long sequence, and can handle extremely long sequence, makes the inference process more efficient. On the inference scenario, we have further done a lot of optimization, including computation and memory optimization and data preprocessing parallel optimization. For computation optimization, we have rewritten the performance hotspot kernel using Triton. We support faster layer norm and faster attention based on flash attention. For memory, we proposed auto-chunk technology and significantly reduced memory consumption to support inference of sequence over 10,000, covering most of proteins. FastFold also supports a synchronized distributed schedule by using Ray as the engine. Ray is used to schedule each stage of the AlphaFold inference process, parallelizing the data preprocessing part, resulting in at least 3 times the speed of preprocessing. With all these optimizations combined, overall 5 times speed up can be achieved. In conclusion, FastFold systematically addressed the challenges of AlphaFold training and inference, and proposed several optimizations that significantly reduce the time and cost of training and inference. This makes AlphaFold more accessible and effective. With FastFold, it is possible to design and deploy more effective protein structure prediction models, and also to create a larger model for improved performance.